So stomach is one of the main sites of mechanical digestion, but also chemical as well. What's going to happen is the stomach, we're gonna have food come in from our cardiac sphincter and dropped into the stomach. The first part of the stomach is going to be called the cardiac region of the stomach. The next section is going to be called the fundus, and then the final section will become, before it hits this turn, is going to be the pyloric region of the stomach. And you can see another sphincter right here called the pyloric sphincter. Food will typically, depending on what you're eating, will sit in the stomach for about 4 hours. And that was a terrible word I used, sit, because it definitely isn't sitting. It's constantly being moved around and churned around almost like a cement mixer. And these little folds that are in the stomach are called rugae. And these rugae are designed to help act like baffles. So food hit them and sort of start to break down into small components. When food is sort of liquefied to a point that we know that we can sort of push it through, we're going to go ahead and take that food and start to move it down the small intestines. In the stomach, we go ahead and turn that food into what we're going to call chyme. And chyme is going to be that, remember that bolus that was coming down of food? Now we've sort of liquefied it, almost like if you take food and you put it in a blender and it turns into this pulpy mess. That's what chyme is going to look like. If you've ever puked a couple of hours after you ate something, you're looking at stomach acid and chyme. The stomach can stretch to about 1 gallon. And really it doesn't absorb nutrients. That's sort of a misnomer. It does a little bit of lipids. Any drugs that are lipid soluble it can go ahead and absorb, but that's pretty much it. The stomach has a couple of mucosal layers and things, but they're really designed to have a thin mucosal layer that sits at the end of the stomach. You can see the folds. Those are going to be those baffles that we talked about. And what we're going to do here is we're going to go ahead and break down some of these gastric pits. The gastric pits, which were the little folds that you could sort of see in the stomach, has a couple things going on. We have a mucosal neck, and that's going to basically be the area that mucus will come out, acids and mucus will kind of come out here and sort of coat the stomach in this mucus, this bicarbonate mucus, to make sure that it doesn't dissolve itself. We're going to have these parietal cells. I guess here they're going to be the blue ones, and they're going to secrete out HCl, hydrochloric acid, and that's going to start to rise to the top and sort of sit in the stomach. That's where the acids come from. All the way at the end of our gastric pits, we're going to have these things called chief cells, and they're going to produce this thing called pepsinogen, and that's the inactive form of pepsin. It's also going to produce this thing called lipase. You could tell right away it's an enzyme, and it's designed to absorb fat, break fat down. And what we're going to do here is pepsinogen, as it comes out of the chief cells, eventually is going to hit that HCl. And when it hits that HCl, it's going to go from pepsinogen, the inactive form, to pepsin, which is the active form. And pepsin is designed to break down peptide bonds, and it's going to start to facilitate those proteins into becoming smaller proteins, and eventually when we get them into our small intestines, we're going to break them down to amino acids. It's a very difficult job, and we've got a lot of things designed to help us break proteins down. And then finally, we're going to have these other cells that are going to happen in the pancreas that's going to help make this thing called gastrin, in the stomach. And they're a hormone called gastrin, and that's going to activate the process of the stomach moving. And we'll talk about what gastrin does soon. Pepsin is going to be the active form, and that is going to move and break things down. We know that pepsinogen only turns into pepsin when it hits HCl. and it's the active form that's going to help break proteins down. We don't want to do that down here. That's why we don't go ahead and just make pepsin, because if we did, we would burn a hole through the stomach and get an ulcer. But by having the pepsinogen come out of these pits and then hit some HCl outside the stomach, it'll go from the inactive form, the pepsinogen, to the active form, pepsin, and that's where it's going to go ahead and start breaking things down. The functions of HCl is going to be to drop the pH of food to about 2 to 4. This is awesome when it comes to killing pathogens. Those pathogens like a pH of 7, and to have a pH of 2 to 4 is immediately going to kill them. Also helps break down some plant cells so we can get inside of them. Also helps break iron into something that we can go ahead and ingest and move to the rest of the body. The intrinsic factors that are produced in this process are going to be the parietal cells. They're going to go ahead and help not just go ahead and make the HCl, but they're going to go ahead and aid in bringing in some vitamins, especially B12. B12 is used for so many things, helping to make red blood cells, brain and nerve functions, memory, DNA synthesis. They're a precursor for that NAD and FAD. So all those things are designed from that. The stomach lining itself is going to be covered in mucus, and it's a mucus that has a bicarbonate in it, and it's designed so those enzymes that are designed to break down food don't break down your stomach. Because I know this sounds gross, but you could go ahead and take a stomach of an animal and eat it, and of course your stomach is going to break that down. So your stomach is going to break down the stomach. But because your stomach is covered in this gastric, mucosal lining, this mucosal membrane that has this bicarbonate in it, it protects your stomach. And of course, we talked about the sphincters. We talked about the pyloric sphincter, and then at the top of the esophagus to the cardiac region is going to be the cardiac sphincter. We talked about the mucus layer that protects the stomach, and that's going to be made of a bicarbonate. We talked about the stomach being made of tight junctions, and that prevents leakage of the gastric juices into the body. Could you imagine if those gastric juices leaked into your body? They would start to break your internal organs down. If you had the ability to spit this gastric juices out without neutralizing it, like from your mouth, you'd be able to dissolve tissue. If you spit it out onto a car, you'd be able to peel the paint off of it. That's how low that gastric juice's pH is. Because of that, it does damage a lot of those epithelial cells, and it causes them to sort of degrade and stop working. So we have to constantly be making them. Gastric mobility happens through a couple of different phases. It takes about 4 hours or so to get food to go from the stomach, slowly bring small amounts of that chyme. Remember, it starts out as a bolus, gets into your stomach, we call it chyme, and then eventually we dump it into your stomach. And that takes about 4 hours. We dump in about 3 mils at a time into your small intestines. That's why you can see the pyloric sphincter has almost like this little mini cup in there, and it's designed to sort of, as soon as it fills, the pyloric sphincter closes, that food dribbles in. A couple minutes later, it's going to open up again to allow us a little amount of food to come in, of chyme to go in, and sort of drop it in again. We have a definite nerve factors that go ahead and help move the stomach, and most of them are going to be through the vagus nerve. They're definitely the sympathetic system that does this. And our 3 regulatory systems are going to be, the first one is going to be the cephalic phase, and that's going to be stimulated by you seeing food, smelling food, hearing food, and that's going to travel down your vagus nerve and tell your body to start producing hydrochloric acid and that pepsinogen, which will turn into pepsin. And it's also going to start producing gastrin, and gastrin is going to help make the liquids start to come out, and even the stomach start to move. Phase 2 is going to be the gastric secretion phase or the gastric phase. And that's going to allow food to come into the stomach. The stomach is definitely going to be allowed to stretch. This is where we're going to go ahead and definitely make those pepsinogen to start to break down those protein peptides. We're going to definitely produce a lot more acid now, and all of these things are going to be stimulated by ACH. Remember, that was the thing that sort of helps with muscles. We're also going to make that gastrin, which is a hormone, and it's going to trigger secretion of a little more HCl. So we had a little bit to sort of start the system, but now that we can sort of figure out how much food you ate based off of how much the stomach is stretching, we'll make more HCl to sort of compensate. If you only ate 2 or 3 grapes, we're not going to fill your whole stomach with HCl. That's a waste of chemicals, waste of time, it's just stupid. But if you ate a whole sandwich or a sub to yourself in this, terrible thing that you did, you're going to go ahead and now fill up your stomach with HCl, and the trigger for how much HCl you need is by how much your stomach is sort of stretch open. Finally, you're going to have this regulation of the gastric secretion, which is our last one, and that is going to be 2 phases. The first one is going to be where we're going to start to make that chyme and start to break that food down. This is going to make the stomach start to spin. We're going to make sure that mucosal cells start to produce that gastrin to get the stomach moving. We're going to go ahead and release a lot more HCl. And then we're going to do the inhibitory phase, which is where we're going to stop the process. So you could see that there's a little bit of overlap here from the regulatory phase. It's going to happen from the gastric phase even here. But then, of course, the stopping part will be this last phase. And this is where we're going to start to move food into the small intestines. We're going to go ahead and start triggering the release of a couple more hormones that we'll talk about soon, and they're really all designed to break proteins down. It's so hard to break proteins down that we have a lot of chemicals designed to do that, and one of the last slides in here really does focus in on all the different chemicals designed to break protein down. Let's go look at that one. You can see just from a couple, we have in the stomach, we have pepsinogen and pepsin, excuse me. So pepsinogen is the deactivated form. And then our next one is going to be our trypsin, which has a precursor to the trypsinogen. And trypsin is basically designed to go ahead and break down those peptide bonds. We are also going to do most of this in the brush border enzymes and make that process happen. And as soon as we do that, we're going to go ahead and have those proteins. So we have a little bit happen in the stomach. That's going to be pepsin. Trypsin is going to happen in the small intestines, and we're also going to make pepsidase. And all of these are designed to break proteins down to its smallest component. Pepsin is going to make this protein go from big to small. Trypsin is going to make that protein go from a little smaller, and then finally pepsidase is going to be able to break these down to the final amino acids. All these 3 have to happen in unison to sort of help make those proteins go from your stomach, your small intestines, excuse me, eventually into your bloodstream.